Hello everyone and welcome. I'm the host of today's meeting, Jessica Tu, from GenScript ProBio Marketing Department. On behalf of GenScript ProBio, I'd like to thank you all for attending today's webinar. GenScript ProBio, a subsidiary of GenScript Biotech Corporation, offers end-to-end CDMO services from drug discovery to commercialization with proactive strategies, professional solutions, and efficient processes in CGT, antibody, and recombinant protein drug, aiming to accelerate drug development for customers. Today's webinar will include one talk. It will take 45 minutes for the presentation and 15 minutes for the Q&A session. If you have any questions, you can submit at any point during the webinar using the chat or Q&A panel situated in the menu on the right. Now, I'm delighted to introduce today's speaker, Rubin Jiang, Director of Antibody Process Development and MSAT Department, GenScript ProBio. After receiving his master's degree in biotechnology from Northeastern University in 2014, he worked at Sanofi Genzyme as a process engineer for upstream process development of antibodies and recombinant proteins. From 2014 to 2020, he worked in the process development department of Merck Sharp & Dohme as a researcher and senior researcher, responsible for the upstream process development, process characterization, and validation, as well as leading the upstream process development of Merck Sharp & Dohme's continuous production and process online analysis upstream development. During this period, he participated in the publication of 7 academic articles. From mid-2020 to 2022, he worked in Hangzhou Yanji Shi as Deputy Director and Director, responsible for Upstream Process Development Department, in charge of CDMO upstream process development, and also promoted an enhanced fed batch process and continuous perfusion process platform. During today's webinar, Mr. Rubin will be discussing cost reduction strategies in clinical stage process optimization for biologics. Now, I'm delighted to hand over to today's speaker, Rubin Jiang.
Many thanks for the introduction. And today I'm going to, the topic is about cost reduction strategies in clinical stage process optimization for biologics. So after your product got into the IND approval and the product start in clinical, the next question we have to ask is, when I'm going to do the manufacturing, and if I'm going to do the commercial manufacturing, how could I reduce the cost of good for this specific product, so this product can have price advantage in the future competition? Today we're going to answer this question. There are 4 topics I'm going to talk about. The first one is about fact sheet. We're talking about give you a brief introduction about ProBio and our track record. The second topic will be the life cycle management for clinical stage CMC process. The third topic is the cost reduction strategies, and the fourth one is we will have a case studies for later stage process development, which happened in ProBio. Just a quick introduction. So far, ProBio got 38 global INDs, including from China, Korea, EMA, and FDA. So we have 100% IND at one-time pass. And the most advanced project already got the emergency usage application. And we have 2 marketed medical devices product, and ProBio was antibody supplier for these 2 products. So far, we experienced more than 180 cell line development projects, and we have experienced 90 CMC and CMO projects. 60% of the projects are from US, EU, and AP. And we have manufactured more than 120 GMP batches successfully. And our service, starting from the discovery and including preclinical development, early clinical development, and also the late stage clinical and commercialization service. Just a quick introduction about our cell line. Our TRIL cell line can reach on average for antibody 5.84 gram per liter for antibody, and for bispecific antibody, the titer is average at 4.84, and the proteins titer average at 4.18. And we provide 60 PDL stability for all our cell line. And as the graph showed, at PDL 43, you can reach 2,000 liter, and with PDL 60, you can scale up more than 8,000 liter scale. And in the middle, the table actually showing the scalability and the stability of our cell line. You can see between 3 liter and 2,000 liter ROM, the titer are comparable. One is 8.9 and another one is 8.47. In the product quality attributes, especially the purity attributes, are pretty much comparable. And if you look at the right middle, is a titer distribution. You can see more than 80% of our MAP process reached between 4 to 8 gram per liter. And there are 10% of our MAP product is between 3 and 4 gram, and none of our project ever go below 3 gram per liter. And ProBio is proud to deliver many complex molecules. For example, a big section of our projects are from bispecific antibody section. We have IgG-like or FC-related antibodies like symmetric bispecific antibody, including IgG SCFV, or IgG with a subdomain antibody, or asymmetric bispecific antibody, for example, IgG with the cytokines or trispecific antibody. And we also delivered quite some projects for non-IgG-like projects. For example, it's Fab, single-chain FV, or single-chain FV connect with each other, or single-chain FV connect with Fab. On the other side, another big portion of our projects are from IgG fusion protein. They have a protein connected with VHH, or protein connected with SCFV, or we have FC fusion protein with protein with FC. And the third session of our projects are also from recombinant proteins, for example, vaccines, microglobins, enzymes, and factors. In terms of the platform, so far, we have our platform process summarized from nearly 100 ProBio CMC projects. And in the same time, in terms of the manufacturing capacity, we have 25 liter waves, 3 50-liter bioreactors in GMP facility, 7 200-liter, 5 500-liter bioreactors, and 8 2,000-liter bioreactors in the GMP facility. We also have ATF4 and ATF10 to support up to 300-liter perfusion batches. All these bioreactors, they provide flexible manufacturing scale and flexible manufacturing schedule for our clients to meet the clinical requirement. And for the pilot scale and the commercial scale, we also have our filling line with multiple formulation available. For example, we have a 2 ml all the way to 20 ml filling volume, and we can handle pre-filled syringe and also the nasal spray. About our platform, so after we deliver so many different projects, we actually accumulate all our experience in 3 different platforms. taking monoclonal antibody on the top, for example, for cell line, we still use the CHOK1 cell line with our patent up vector, which can greatly increase the titer. For upstream process development, for monoclonal antibody, we just use fed batch process. If we really need high titer, for example, 8 or 10 gram per liter, sometimes we want 15 gram per liter titer, then we will go to intensified fed batch process. Or if we want to greatly reduce the cost of good and also in a smaller facility, we can use perfusion process. For downstream, for monoclonal antibody, we usually just classic 3-step purification step. And for formulation, we usually deliver the project in liquid. And also provide in-use stability study to help better application in clinical trials. For bispecific antibody projects, it's a bit different in cell line session. Besides, we have our CHO genes cell line and the up vectors. There is one special step we will do is plasmid ratio testing, which means for different project, we may separate the molecule into different plasmid and inject to the cells. However, by adjusting the plasmid ratio, we can greatly reduce the aggregation and also the mismatching in the bispecific antibody production. And for upstream, for bispecific antibody, again, we use fed batch, intensified fed batch, or perfusion process. And for downstream, for bispecific antibody, multiple affinity ligand might be tried and used. This is really depending on your target product. In the same time, we will use the mixed mode chromatography to better separate the homodimer and our target product. And for formulation development, really depending on the stability of the target product, we may use the liquid or lyophilized powder for the formulation. And again, we provide in-use stability study to support clinical trial application. For recombinant proteins, again, we use CHOK1 genes and up vectors. In this case, up vectors can greatly increase the recombinant proteins expression level and bring them to more than 5 gram per liter level. And for recombinant proteins, there is no affinity column available, so we will have to try multiple capture modes. In the same time, always mixed mode chromatography or hydrophobic chromatography is used. And because we have experienced some of those blood factor projects, we also have a platform for unstable molecules in our process. So we can handle those unstable products, not only in the process development stage, but also in the large-scale manufacturing stage. Again, for recombinant proteins, the formulation development including liquid or lyophilized powder, and we can also do the in-use stability to support the clinical trials. And for recombinant protein vaccines, we also provide immunologic advanced tests for our clients. After all those difficult projects, in order to deliver our projects in a standard way, scientists in ProBio actually accumulate all the knowledge and the practical experience into different sessions, and we call that ProBox. When with this ProBox, our scientists can go into the database and find out if there is similar difficulties happened before and what would be the previous solution and what is the principle of this problem, how to solve it, and also what the expectation outcome of this procedure. So the entire database actually including cell line, cell culture, purification, formulation, and analysis. Take purification, for example. People may have aggregation homodimer removal problems, and if we clicked into that this problem, and there are quite different solutions. For example, variable gradients pH elution based on affinity difference, or variable gradients pH elution based on charge difference, or we can also do basing on the hydrophobicity differences or multi-characterization separation based on the multi-model chromatography. And if we further click this one, there will be the case study and also the expectation outcome of this project. After the introduction, I'm going to go into today's main topic about how to reduce the cost reduction strategies for clinical CMC projects. First, let's talk about the life cycle management. Here, I'm showing a life cycle starting from the discovery all the way to the post-marketing project. You can see after the preclinical studies and the product will go into the IND phase. Then phase 1 is for safety study, phase 2 is for efficacy, and the phase 3 is for safety and efficacy. After it's approved, again, it requires continuous monitoring, including safety, efficacy, and consistency. And usually, after the discovery, ProBio as a CDMO will jump into the project and we provide the toxicology product to our clients, so the tox study will start. During the IND, we will summarize our produced GMP level material and also help our clients to fill in the IND application. Once we got into the clinical trials, for manufacturing, at the end of the phase 1, usually we recommend to do the process improvement. And by the end of the phase 2, we do recommend our clients to have large-scale or commercial-like manufacturing process to produce the material which we'll use for phase 3. Okay, and in terms of how and when we should do early stage and late stage process development, I'm going to give you a quick analysis. In this stage, in the table in the middle, I'm showing the success rate for clinical trials at different stage. For preclinical, the successful rate is around 70%. In phase 1, it's only 54%. In phase 2, it is the lowest one, it's at 34%, and at phase 3, we have 70% successful rate, and at the registration, we have 91% rate, and overall, it's around 10%. It's slightly higher than 10%. Then people will start to ask, after the IND approval, when should I do the late stage process development, and when should I start the PC and process characterization and process validation? Here, in the table, I'm going to give you a quick analysis. For example, if after the IND's approval and the project in phase 1, usually we can do late stage process development at phase 1. The advantage is once your project have a faster track or is on a faster track, then if you did your late stage process development in phase 1, then the project can go into the process characterization and the process validation very quickly without any CMC problem. And in this case, so that you will before you enter in the phase 3 process, you will have your CMC process updated with a higher titer, and also the CMC material can be produced from a similar scale as your commercial scale, which is recommended by the regulatory agencies around the world. However, there will be concern. If the process clinical trial failed in phase 1, all the late stage work you've done in phase 1 will go to drain because the process failed. So there's a high sunk cost if the phase 1 trial failed. Then what would be the advantage of start our late stage process development at phase 2? Of course, the pros is there's no sunk cost if the phase 1 trial failed. However, because it takes at least 9 months for the process optimization and the manufacturing and the supplemental application, if your process if your clinical trial is in a fast track, then the clinical trial might have to be slowed down in order to wait for the CMC session to be ready. So this is something most of the company wouldn't want to see, and especially for if we're working in the CMC area, the best scenario is always the CMC is ready and waiting for clinical trials data. However, if the CMC clinical trial data is good and ready, waiting for CMC, that will be a very urgent scenario for CMC section. And of course, we don't recommend to do any process change during the phase 3 or in your phase 3 clinical trials, because this will trigger additional clinical trials to verify the safety and the efficacy of the new process for your product. Here I'm showing a very overview timeline for late stage process development. Starting from the first 2 months is technical transfer, and after we have the cell and the clone and your process, we will start process optimization. After the process optimization, we will have a 2,000 liter GMP run. This run is responsible to supply the clinical material for phase 3 or phase 2 or phase 3 clinical trials. In the same time, right after we have the 2,000 liter GMP materials ready, there will be work to submit the supplemental application for EMA and FDA. So the material produced from the 2,000 liter GMP environment can used for the clinical trials. After this, we will start our process characterization process, which generally takes 6 months, including risk assessment, scale-down model, building a scale-down model. Then we do the DOE studies, and after we have the DOE studies and the potential critical process parameters, and we will scale down to the worst scenario, and which is called the worst case study. If this worst case study works, then we will have our range successfully defined for all our critical process parameters. And after we have the CPP, which is critical process parameters, the process can move into the process validation stage, which is in 2,000 liter. And after 3 2,000 liter validations, the project will be ready for the BLA. Well, for a CMC session, it will be ready for the BLA. So altogether, including technical transfer, clinical material production, and also the PC and the PV, it takes 9 plus 14 months. And one big question for CMC session need to worry about is the cost of good for the commercial manufacturing process. And let's talk about this. In ProBio, we actually built a cost of good model in order to help our clients to assess the cost of good in the future commercial manufacturing scale. And by defining, in the general definition, the cost of good, including the fixed assets, including your equipment, facilities, and operating expenses, raw materials, and consumables. However, in ProBio's cost of good model, which I'm going to show you in later slides, we're actually basing on a real manufacturing scenario, which including the raw materials, equipment, facilities, and quality control, and the service costs. In this case, the equipment and facilities is actually included in our service cost. So because when clients come to us, clients wouldn't pay for the facility and the equipment. So this actually, in this case, that's a different part for the ProBio's cost of good model and the general cost of good model. And according to the ProBio's model, the factors that impact the commercial process cost of good are ranked like cell culture productivity increase actually have the biggest impact if you want to reduce your cost of good for your product. And the second is scale up. Scaling up from 500 liter to 2,000 liter or even 6,000 liter have a really big impact to the cost of good. The third one would be the raw material replacement. In this case, upstream raw material replacement really play a role in general process, because for resins and columns and resins, those material can be reused. So in a commercial manufacturing scenario, the cost of the raw materials for downstream is really low. However, for upstream, especially the media and also single-use bioreactors and also the depth filtration, single-use depth filtration equipment, they are all single-used. So which means every time there will be new media and a new filter and a new bioreactor. So raw material replacement in upstream will have a bigger impact to the project. Here I'm showing you the improved cell culture actually can improve the cell culture process productivity, can actually reduce the cost of good. On the left picture, you can see if starting we have a 3 gram per liter process, and if we increase the titer to around 4 gram per liter, 6 gram per liter, the cost of good will be reduced by around 40% when the titer actually increased by 100%. By optimize the parameter optimization and medium optimization, and also adjust the inoculation cell density, we actually can achieve the process increase in the cell culture process. In the table on the bottom is actually showing a really nice case, which original process was only 3.9 gram per liter, harvest viability is 81, and after the optimization from the media, the titer actually increased from 3.9 to 7.6, and the harvest viability was comparable compared with the original process. In the same time, the product quality attributes are similar with each other. In this case, we only showing the purity product quality attributes because this is all other informations are confidential, but they are all comparable. Here at ProBio, we actually also provide a service for titer increase through N-1 perfusion. By doing this, we actually increase the inoculation cell density starting from the 0.5 million cells all the way to 10 million cells. And by doing this, we usually achieve around 100% increase in terms of the titer. So through this N-1 perfusion process, we actually tested more than 15 different projects, and the majority of the project can actually reach the 100% increase, and some of the project even tripled the titer at the end of the process. And for one of our process, for N-1 perfusion, you can see the titer actually comparable between the fed batch 3 liter intensified fed batch process and the 500 liter intensified fed batch process, which means the process is pretty robust across different scale. And the N-1 perfusion process wouldn't impact any quality attributes for the target product. In this case, we actually put the Amber 15 fed batch process, 3 liter intensified fed batch process, and the process material from 500 liter. And you can see the product quality attributes are pretty comparable. Scale up is another big factor to reduce the cost of good. And on the left table graph, I'm actually showing the cost of good reduction when comparing with a 500 liter scale. 500 liter scale is set at 100%, and when the scale goes to 2000 liter, the cost of good was reduced almost 50%. And further increase the titer scale will also decrease the cost of good percentage. In CDMO-based model, the depreciation and the labor costs are actually excluded, because in general, if it's a manufacturing, if this is a pharmaceutical company's scale and the model, people will argue, because I have a huge facility. Let's say I have a 6 1,000-liter bioreactor facility, and they were also considering the depreciation and also the labor cost of that facility, and also the utilization rate for the facility. But in the CDMO-based model, the depreciation, labor cost, and also the facility utilization rate is actually excluded. That's one of the advantages of using the CDMO organization. Again, if the cost of good of 400-liter scale was only around 30% of the cost of good of the 500-liter scale. And in ProBio, we actually provide 8 times 2,000-liter disposable cell culture system, which will actually allow us to have a maximum production volume around 6,000 liter in scale out mode. We can actually effectively reducing the production costs. However, in terms of the scale, it's not as big as possible because the scale for the truth is also have to depend on the product demand, storage, stability, and also the upfront cost should be also considered when we're choosing the scale. Here I'm actually showing you a map of the GMP facility of the ProBio. On the left bottom is our design. Right now we have upstream production 1A and also the purification facility ready for the phase 1A, which is now operating now. And we also have the cell culture and purification area for the phase 1B, which will further have a 2,000-liter bioreactor, and the downstream area is also reserved. Once we have the production 1A filled up, and we will start the phase 1, use the phase 1B for the other clients. In the same time, in the commercial manufacturing scale, we also have our fill and finish line, which meets the FDA, EMA, and the NMA regulatory requirements. We have a freeze thaw machine, which means we can deal with large scale DS material. In the same time, we can also deal with those temperature sensitive or freeze thaw sensitive molecules with this freeze thaw machine. And we have filling line from both, and also we have a lyophilizer and a visual inspector for this fill and finish line. This fill finish line is also operating by now. The third big factor for the cost reduction is the raw material replacement. The raw material for biopharmaceuticals industry are pretty expensive, and sometimes they can account for around 30 to 40% of the total production costs. So in our commercial manufacturing or late-stage process development, one of our tasks is actually to use the lower priced raw materials, especially for commercial manufacturing scale. For commercial future or going to the commercial manufacturing, you don't really want to use commercial raw material because they are quite expensive. However, our goal is to use lower priced raw material in the same time without impacting the productivity and product quality. For example, the ones we can potentially swap is cell culture medium, resins, and the vials. They are pretty expensive and they are easy to swap. Of course, we will also select our backup raw material vendors to test in our late stage process to make sure they're replaceable, to prepare for any emergency, and also to prepare to have a better supply chain robustness. In the table below, I'm showing a quick example. This is one of the process which is doing the late stage process development in ProBio. You can see in the portion of the raw material cost at a different process session, the cell culture material media actually account for around 50 to 65% in the upstream total cost. For the downstream total cost, the resin actually account for 50 to 77%. And in the fill finish step, vials can actually takes more than 50 to 70% of the portion of the total cost. And for those, we can also find lower priced materials. For cell culture media, the lower priced raw material we recommend to our clients are actually only 30% of the original cell culture media we use. And for the resins, we also have a robust brand that can account for only 75% of the original cost for the resin. And for vials, we find because our commercial manufacturing have a capability of cleaning and sterilize the vials, we can use the vials without the pre-sterilization. So the new vials are only 60% of the original vials we use. So at the beginning, before the raw material swap, all the raw material will cost for about 30 to 40% of the total cost of a CMC. After the swap, the proportion of the raw material cost will decrease to around 11 to 20%. And here, in terms of the raw material, I would also recommend one of our uphold medium, which developed in ProBio in-house. And here we have our uphold media, and with two branding A and brand B. They're both from the global famous vendors for their media. And we can see at different project we tested, our in-house media always have a higher or comparable titer compared with those global famous branding media. And also our media have a very consistent performance, and we actually used 3 different powder batch and do the cell culture, and the culture performance are quite comparable with each other. And our in-house media are chemical defined media with no chemical animal components, and we also have basal media, feed media, and the perfusion media for our in-house media. And it's proven to have a good cell culture process, cell culture growth, and they have a good productivity, and also the product quality are also quite comparable. Of course, in our in-house media, the trace element might a little bit different from other brand media. So that one, we will have to address the problem in our late stage process development. And by average, our media can improve the titer by around 45%, 49%. And we have the media have a robust manufacturing process and a good batch-to-batch consistency. Here is actually one of the case that our media is used in the GMP environment. The 3 batch are quite comparable from the 3 liter to the 200 liter process. And you can see the viability is also quite comparable with each other. And in 3 liter, the titer was 9.6, and in 200 liter, the titer was 9.17. And all the 3 runs have a very comparable product quality attributes. This is a bispecific project, and the titer reached 9 grams per liter, which is pretty very promising. After the process modification and process improvement, another big factor for the CMC people have to worry about is a supplemental application. Here I'm going to give you some guidance. For process modification and preclinical stage, the most important thing is comparable. The new process have to be comparable between the materials for tox study and for the clinical studies, which means after your tox study, you may modify your process, and before you're going to the clinical trials, regulatory agency will worry about if there's any difference between your tox study and the clinical studies. And for process modification at early clinical stage, usually at this stage, the regulatory agencies are still worried about the risks associated with the safety. When we are preparing for our supplemental application, we also should address the concerns. For example, if there is external factors introduced to the process, and also what is the impact to the virus and the microbial clarification in the process. If we are going to do the process modification and the material will be used in during the phase 3, and the regulatory agency will concern more because they will worry about process robustness, and we have to show the comparable scale. This scale should be comparable to the commercial scale. And actually, during the phase 3 clinical trials, we don't recommend any change because that actually may impact safety, efficacy, and also the product quality attributes. Any of those product quality attributes change may trigger extra clinical trials. That's why it's not recommended to do the work during the phase 3 clinical trials. But before phase 3 clinical trials is okay. For process modification for marketed product, of course, again, we have to answer the question like a safety, efficacy, and quality. Make sure they're not impact during the process optimization. The last but not least, I'm going to do share some case study for the late stage process development. So first example is for late stage process development. So this is a recombinant protein with an unstable process and a large bioassay variability. So this is actually a broad factor project, which means the product is very sensitive to the environment, is not very stable in room temperature and in harsh conditions. So this project was tech transferred to ProBio for process optimization and also the phase 2 and phase 3 manufacturing. And this product is also preparing for the BLA. So the process and analytical methods were optimized before the phase 2 and the phase 3 manufacturing. And for the PC and the PV activities were also started after the phase 2 and the phase 3 manufacturing. For the timeline, starting with the technical transfer, then we do the process optimization. After we lock the process, we actually do the clinical manufacturing for the clinical trials. Then we enter the process characterization, and finally the process validation. By the end of the process optimization, we actually greatly improved the process robustness through the process optimization. And also, because there is an urgent timeline for this project, our process characterization was adjusted. So we are able to finish this process characterization within 5 months. This is a quick timeline for the specific project. We have 3 months of time for the process optimization. Then we do the clinical trials, clinical manufacturing, production for clinical trials. Then our RA department will start to file the supplemental applications for this process so they can start the clinical trials in phase 2 and phase 3. In the same time, our PD department started process characterization, which including the risk assessment, scale-down model, building the scale-down model, then do DOE studies and worst case study. Then we have the locked critical product process parameters. In the same time, the clinical manufacturing product will have drug substance characterization and drug substance and drug product stability studies. So the PC and PV activity was finished in 14 months, and before that, there was a 7 months time for the process optimization and the clinical manufacturing. In this case, in different department, we actually have a different improvement. For cell culture process, the cell culture parameters were optimized, including the pH, feeding volume, and also the culture duration, because the viability was improved. So the culture was extended for another 2 days. And we also there was an additive greatly impacting the product quality attributes. However, the formal vendor stopped producing the additives. So the project team actually find backup vendors and to produce these additives to add to the process to make sure the product quality attributes are comparable between the new process and the old process. In this case, we actually identified 2 backup vendors for these additives to make sure the supply chain is robust. At the end of the cell culture process optimization, the titer was increased by 15%. We have a primary and a backup vendors for additives, and also we have a comparable product quality compared with the original process. For downstream, the parameter optimization was also down, and also we replaced some of those expensive raw materials. For downstream, the main results action item is ensure the process robustness. In the same time, by replace the resins, we can greatly reduce the upfront expenses for the BLA activity. And for fill finish, because this product have a really high titer, in the same time very low amount of fill finish, so the fill finish actually have a big impact to the cost of good for this project. The major work we did is replace the vials, which can greatly reduce the fill and the finished product cost. For QC release, our QC department actually collaborated with our analytical development department. They worked together to improve the bioassay robustness. By doing that, we greatly reduced the variability of the potency assay in QC department. And at the end of the process, the cost per vial was actually decreased by 60% after the optimization, and in the same time, by fill finish cost was decreased by 74% alone, and the release and the stability cost was also increased by 52%. Because we actually designed to use a larger scale fill finish line. By doing that, the fill finish and the release and the stability study cost was greatly reduced. In the table on the bottom, I'm actually showing you the process cost compared to the original process. In terms of the material, it's still around 100% compared with the original process. However, and the manufacturing service is actually further increased a little bit. However, the fill finish product is greatly reduced. It was only 26% of the cost compared with the original process, and the release and the stability study cost was also greatly reduced by 50%. This is a second case for the latest stage process development. Our client come to us after they have the IND approval, and they are doing their phase 2 project, and they required a titer improvement. The original titer was only 0.72%. After the cell culture optimization and the feed optimization, the titer was increased from 0.27 all the way to 1.02. And the product quality attributes, as I showed in the table, are pretty comparable. The manufacturing cost was actually decreased by 70% under the same scale. The third one is actually already marketed product. So this product was using a perfusion process. They only have a 5 to 7 mix per liter per day using a perfusion process. Because this is for this process, the original process requires animal components. In the same time, it's a mixture of it's not a chemical defined media. So in ProBio, we actually rebuilt the cell culture. In the same time, rebuilt the cell line. In the same time, we use our screening the cell culture media, which have the good quality attributes with this product. And we also optimize the downstream process. By doing that, the titer was increased more than 10 times, because it's not very comparable, because previously they're using the perfusion, and right now we're using a fed batch process, which is a lot easier. The process complexity was previously is complex and rigid. Using the new process we developed, it is simple and flexible. And we're actually helping our clients to discussion with the regulatory agency and to see the improvement and the potential replacement for the marketed product. And that's pretty much for all the content for today. Again, in a summary, efforts in late-stage process development primarily focus on the process robustness, cost reduction, and the preparation for the commercial production. And ProBio actually possesses a rich experience in the late-stage process development and process characterization and also process validation. So ProBio will help our partners in contributing to human health. That's all I have for today. And you're welcome. If there's any questions, it's a time to share. Thank you.
Yeah, thank you. Thank you for Rubin's excellent presentation. If you have any questions, please type it on the chat or Q&A panel. As you enter the questions, you can also follow our LinkedIn account, GenScript Profile, and Korean audience can also scan this QR code and follow our Kakao channel. There will be more online sharing resources, and we will share them on our LinkedIn and Kakao channel at the first time. We received some questions from the audience. Let's take a look at the first question. What would be the best strategy to minimize the costs for the late stage and commercial manufacturing process? Rubin, could you please answer this question?
Oh, thank you. So in summary, I think if you want to reduce the commercial manufacturing cost, first of all, you want to have a really productive process. You already have your cell line, but you can improve the titer through media optimization, and also if downstream there's a recovery problem, you may also have to work with the recovery problem to improve the recovery rate. On the other side, it's always recommended to use a higher scale if you want to reduce the cost. Yep, that's pretty much increase your productivity and also increase your scale. In the same time, if there are expensive raw material used in your process, you may have to consider to swap it to reduce your cost of good.
Okay,
Thank you.
Thank you. Thank you for your answer. And the next question is, in general, what would be the risks associated with process change, scale up, and technical transfer between different facilities?
I'm sorry, Jessica, could you repeat your question?
Yeah, I can repeat it. In general, what would be the risks associated with process change, scale up, and technical transfer between different facilities?
Oh, yes. So this is actually a very common question when clients sometimes concern if it's okay to, move one of the if it's okay to remove from one CDMO to another CDMO, or if I work with you, could I potentially transfer the process to my own manufacturing facility after the clinical phase? Yes. So there are several guidance you have to follow, and the most important thing is probably the process product quality comparable for your process for both different end. For now, for upstream is pretty comparable because nowadays, if you are using the single-use bioreactors, the performance between different types of the bioreactors with the fed batch process are pretty much the same. You wouldn't see the difference. However, for the downstream, it might be harder because each facility, they may have their own downstream design, and there might be gentle difference. But those usually wouldn't pose any risk for impacting your product quality attributes. And I think one of the most important thing is you have a really good comparability study protocol and also a strong analytical department, so they actually can handle all those comparability studies and by doing that, if you have your comparability studies and you have your product quality attributes comparable between different manufacturing facility, the supplemental application for your clinical materials will be relatively easy.
Yeah,
Yep, very nice. Thank you for the question.
Yeah, thank you. I also see a question from the chat room. There is a question about the uphold media. Is it a suitable media for the CLD development, cell line development? For example, a mean for stable protein production from a stable clone. Could it be a production medium?
Yes. So for the uphold media is actually consists of basal media and the feed media. In our platform process, we actually have the choice that we can use the uphold media all the way starting from the transfection and to the clone screening and also pool selection and all the way to the cell culture process. So uphold media is actually was developed with our internal in-house cell line, so they actually feed with each other very well. And basing on our observation, with the same cell with our cells, is always our media always performs better than the commercial medias.
Thank you.
Yep,
Thank you
a
for your
lot for the
answer.
question. Yep.
Yeah. That's all the questions I received by now. If there's no more questions, thank you all for your time today.
All right. Thank you everyone for attending this webinar.
Yeah, thank you. And we are also going to hold the CGT offline seminar in South Korea on 23rd May and Biologics seminar on 5th July. If you are interested in joining it, please kindly fill in the survey through the link we sent in the chat room. We are glad to meet you there. If you have any questions, please contact us through our official email. Let me join others in thanking Rubin Zhang for his time today. And thank you all for attending today's webinar. Look forward to meeting you again soon. Thank you. Thank you. Bye-bye.